Your skin replaces itself roughly every 27 days. But what happens at the cellular level, underneath the surface you can actually see, is a process far more precise and far less understood by most people. There's a biological mechanism that runs quietly inside your skin cells, one that researchers have been studying with increasing urgency over the last 2 decades. And when it runs for 72 hours straight, the changes happening inside each individual cell are not small. The outermost layer of your skin, the stratum corneum, is composed entirely of dead cells called corneocytes. These are keratinocytes that have completed their life cycle. They've lost their nucleus, flattened out, and packed themselves with keratin to form a physical barrier between your body and the external environment. What you're looking at when you look at your own skin is essentially a carefully organized layer of cellular remains. The living activity is happening much deeper. Beneath that outer layer, in the stratum basale and stratum spinosum, keratinocytes are actively dividing and moving upward through the layers of the epidermis in a process called epidermal turnover. As they migrate, they undergo a programmed transformation, changing shape, producing structural proteins, and eventually undergoing a specialized form of cell death called cornification. This entire journey from the base of the epidermis to the surface takes approximately 2 to 4 weeks, depending on age, skin condition, and other biological variables documented in dermatological literature. Inside these living keratinocytes, autophagy operates as a quality control system. A 2019 review by Eckhart, Schachler, and Gruber, published in Frontiers in Cell and Developmental Biology, described autophagy as critically involved in skin homeostasis. It degrades damaged organelles, misfolded proteins, and intracellular pathogens, recycling their components back into usable molecular building blocks. Without this process functioning properly, cellular debris accumulates, and the consequences extend well beyond what any surface-level observation can detect. Autophagy, from the Greek meaning self-eating, is a lysosomal degradation pathway that cells use to break down and recycle their own damaged or unnecessary components. In skin cells specifically, this process targets a range of structures: dysfunctional mitochondria, aggregated proteins, lipid droplets, and even fragments of the endoplasmic reticulum when it becomes stressed. The machinery responsible for this involves a family of proteins called ATGs, or autophagy-related genes, first mapped systematically by Yoshinori Osumi, whose work earned him the Nobel Prize in Physiology or Medicine in 2016. In keratinocytes, autophagy plays a particularly layered role because these cells must survive long enough to complete their migration through the epidermal layers while simultaneously managing the oxidative stress that comes with UV exposure and environmental contact. Research published in the Journal of Investigative Dermatology has shown that autophagy in keratinocytes helps regulate the degradation of melanosomes, the organelles that carry melanin pigment, which directly connects this cellular process to how pigmentation is distributed across the skin. Fibroblasts, the cells in the dermis responsible for producing collagen and elastin, are equally dependent on autophagy for their function. Research by Tashiro and colleagues, published in Biochemical and Biophysical Research Communications, 2014, demonstrated that impaired autophagic flux in dermal fibroblasts from older donors correlates with alterations in extracellular matrix protein content and deterioration of dermal integrity. The skin, in other words, is not a passive structure. It is an organ whose quality depends substantially on the intracellular recycling capacity of its constituent cells. Autophagy is not a switch that flips on at a single moment. It operates at a basal level continuously, but its activity increases significantly when cells experience specific stressors, nutrient deprivation being among the most well-documented. Research on the kinetics of autophagic induction has shown that autophagic flux, meaning the actual rate at which material moves through the autophagy pathway and gets degraded, begins to increase measurably within hours of nutrient withdrawal, as documented in studies using both cell culture models and animal models published in journals including Nature Cell Biology. Within the first 24 hours, skin cells begin upregulating the expression of key autophagy regulators, including Beclin-1 and the LC3 protein family. LC3, microtubule-associated protein light chain 3, is one of the most studied markers of active autophagy because it physically associates with the autophagosome membrane during the degradation process. Its conversion from LC3I to LC3-2 is used in laboratory settings as a direct indicator that autophagic activity is increasing. This biochemical shift in skin cells during extended fasting states has been observed in multiple experimental models, though direct human skin biopsy data across 72-hour windows remains an area of ongoing research. What this early phase represents, mechanistically, is a cellular shift in priority. When external nutrient input decreases, the cell begins to extract resources from its own interior, targeting the oldest, most damaged structures first. In skin cells, this means that protein aggregates that had been accumulating, organelles that had been functioning below optimal capacity, and oxidatively damaged lipids begin entering the degradation pathway at an accelerated rate. The skin is not visibly transformed in this window, but the molecular environment inside each cell is measurably different from its baseline state. Collagen is the most abundant protein in the human dermis, accounting for approximately 70 to 80% of the skin's dry weight, according to structural analyses published in dermatological literature. It is produced by fibroblasts and organized into a dense fibrillar network that gives skin its tensile strength and structural integrity. But collagen is not static. Individual collagen fibers accumulate damage over time through oxidative stress, glycation, and UV-induced cross-linking. And when damaged collagen fragments are not efficiently cleared, they interfere with the assembly of new collagen structures. This is one of the documented mechanisms through which autophagic dysfunction contributes to visible skin aging. The connection between autophagy and collagen quality runs through a process called collagen quality control in the endoplasmic reticulum. Fibroblasts synthesize procollagen, a precursor form of collagen, inside the endoplasmic reticulum, where it undergoes extensive folding and modification before being secreted. When this folding process goes wrong and misfolded procollagen accumulates, it triggers ER stress. Research by Forrester and colleagues, published in EMBO Journal, 2019, demonstrated that autophagy selectively clears these misfolded collagen precursors through a selective phagy pathway dependent on the receptor FM134B acting in complex with the chaperone calnexin, preventing the accumulation of dysfunctional collagen fragments and preserving secretory capacity in collagen-producing cells. As autophagic activity increases over an extended period, the dermal environment shifts toward a state with fewer accumulated protein aggregates and less intracellular debris. Research examining fibroblast-specific autophagy, including work on PDL fibroblasts published in Scientific Reports 2021, found that cells with intact autophagic activity maintained collagen synthesis capacity more effectively than autophagy-impaired counterparts, with inhibition of autophagy causing cytosolic collagen accumulation and reduced secretion. The direct implication is that the quality of collagen production in the dermis is partially dependent on how efficiently the cell's internal recycling system is operating, not just on nutrient availability or hormone signaling alone. Melanin is produced by melanocytes, specialized cells located in the stratum basal of the epidermis, and then transferred to surrounding keratinocytes through structures called melanosomes. Once inside keratinocytes, melanosomes are distributed and eventually degraded. The regulation of this degradation process has been shown to involve autophagy. Research published in the Journal of Investigative Dermatology demonstrated that autophagy in keratinocytes participates in melanosome degradation, and that alterations in autophagic activity affect how melanin is processed and how pigmentation patterns develop across the skin. The relationship between autophagy and pigmentation is not straightforward. Studies have shown that autophagy can degrade melanosomes through a pathway sometimes referred to as melanophagy, and that the efficiency of this process influences how evenly pigment is distributed within individual keratinocytes and across the epidermis as a whole. Research by Murase and colleagues, published in Journal of Investigative Dermatology in 2013, specifically examined how autophagic degradation of melanosomes in keratinocytes contributed to skin lightening in experimental models, establishing a mechanistic link between autophagic flux and pigmentation outcome. What this means at the cellular level is that as autophagy operates over an extended period, the processing of melanosomes within keratinocytes continues as part of the broader intracellular recycling activity. The skin's pigmentation biology is, in part, a function of how actively keratinocytes are turning over their intracellular contents. This is a dimension of skin biology that receives considerably less public attention than collagen or hydration, yet it operates through the same core cellular machinery that governs how skin cells manage every other form of internal material. Chronic low-grade inflammation in the skin is associated with a range of conditions, including acne, rosacea, psoriasis, and accelerated photoaging. At the cellular level, this inflammation is driven in part by the accumulation of damaged intracellular material, including dysfunctional mitochondria and oxidized proteins, that activates innate immune signaling pathways within skin cells. One of the most studied of these pathways is the NLRP3 inflammasome, a multi-protein complex that, when activated by cellular damage signals, triggers the production of pro-inflammatory cytokines, including IL-1V and IL-18. Autophagy has been shown to suppress NLRP3 inflammasome activation through multiple mechanisms. By clearing the damaged mitochondria and oxidized lipids that serve as activating signals for the inflammasome, autophagy reduces the substrate available for inflammatory signaling. A study by Nakahira and colleagues published in Nature Immunology, 2011, demonstrated that depletion of autophagic proteins promotes the accumulation of dysfunctional mitochondria and cytosolic mitochondrial DNA, and that this accumulation directly enhances caspase-1 activation and IL-1V and IL-18 secretion, establishing a mechanistic link between autophagic clearance of damaged mitochondria and the suppression of inflammasome-driven inflammation. The scientific literature on skin-specific inflammation consistently positions the intracellular damage load, oxidized proteins, dysfunctional mitochondria, and accumulated molecular debris as a primary driver of the inflammatory tone within keratinocytes and dermal cells. The dermis and epidermis are not isolated from systemic inflammatory signaling, but the local cellular environment plays a substantial role in determining how inflammatory signals are amplified or attenuated at any given point. Autophagy, by continuously reducing that intracellular damage load, operates as one of the primary regulators of that local inflammatory baseline. If any of this changes the way you think about what's actually happening beneath your skin at the cellular level, drop a comment below. A lot of people assume skin aging is mostly about what you put on the surface. The biology tells a very different story. That same accumulation of damaged mitochondria driving the NLRP3 inflammasome discussed above has a second equally important consequence inside skin cells, and it operates through a completely different mechanism. Mitochondria in keratinocytes and fibroblasts influence calcium signaling, apoptosis regulation, and the production of reactive oxygen species, all of which have direct consequences for how skin cells function and age. As mitochondria accumulate damage over time, their membrane potential decreases, their ROS output increases disproportionately, and they begin to function as sources of cellular stress rather than cellular energy. The selective autophagic removal of damaged mitochondria, a process called mitophagy, is one of the primary mechanisms through which cells manage this problem. The Pinken-Parkin pathway is the most extensively studied mitophagy pathway in mammalian cells. When mitochondrial membrane potential drops below a threshold, Pinkon, a kinase that is normally imported into healthy mitochondria and degraded, begins to accumulate on the outer mitochondrial membrane. This accumulation recruits parkin, an E3 ubiquitin ligase, which then tags the damaged mitochondrion for autophagic degradation. Research by Narendra and colleagues, published in Journal of Cell Biology, 2008, and subsequent work in Nature Cell Biology, established the molecular mechanics of this pathway, with later studies confirming its operation in skin-relevant cell types, including fibroblasts and keratinocytes. Over a 72-hour window of elevated autophagic activity, the population of mitochondria within skin cells undergoes selective pressure. The most damaged members of the mitochondrial network are preferentially cleared while functional mitochondria are retained and can continue dividing. The net effect, documented in aging research models, is a shift toward a mitochondrial population with higher average membrane potential and lower average ROS output. Research examining mitophagy in dermal fibroblasts, including work reviewed in Frontiers in Cell and Developmental Biology, 2019, by Eckhart and colleagues, identified mitochondrial quality control through selective autophagy as one of the determinants of how skin cells manage oxidative stress across the aging process. The skin barrier, functionally defined as the stratum corneum and the tight junction network of the living epidermis beneath it, is not simply a physical wall. It is a biochemically active structure whose integrity depends on the precise execution of keratinocyte differentiation. As keratinocytes migrate upward through the epidermal layers, they must produce specific lipids, structural proteins like filaggrin and loricrin, and cornified envelope components in a tightly regulated sequence. Research on autophagy's role in this differentiation process, including studies using ATG7 deficient mice, conducted by Yoshihara and colleagues, and published in the Journal of Dermatological Science, 2015, showed that autophagy-deficient skin grafts displayed reduced immunostaining of keratinization-related proteins, including loricrin, filaggrin, and involucrin, along with acanthosis, hyperkeratosis, and abnormal hair growth. Filaggrin is among the most clinically significant of these barrier proteins. It aggregates keratin filaments during the terminal differentiation of keratinocytes and is subsequently broken down into natural moisturizing factor components, small hygroscopic molecules that maintain hydration in the stratum corneum. Loss-of-function mutations in the filaggrin gene are strongly associated with atopic dermatitis, and the scientific literature on intracellular protein quality control in differentiating keratinocytes, reviewed across multiple studies, indicates that autophagy participates in the processing environment that allows barrier-related proteins to be correctly expressed and incorporated into the cornified envelope. Tight junctions in the stratum granulosum, particularly those involving claudin 1 and occludin, form a second line of barrier defense in the living epidermis, regulating paracellular permeability. Research has shown that the maintenance of tight junction integrity is connected to functional autophagy, with autophagy-impaired keratinocytes showing altered tight junction protein localization and increased paracellular permeability in experimental models. What this body of research collectively establishes is that the skin barrier is not simply a structural outcome of keratinocyte death. It is a functional product of intracellular processes, including autophagy, that must operate correctly throughout the entire differentiation sequence for barrier competence to be achieved. Fibroblasts are the primary synthetic cells of the dermis. They produce collagen, elastin, hyaluronic acid, fibronectin, and a range of growth factors and matrix metalloproteinases that together regulate the composition and remodeling dynamics of the extracellular matrix. Their function is central to the structural quality of skin, and their behavior changes substantially with age, a process linked mechanistically to declining autophagic capacity. Research by Toshiro and colleagues, published in Biochemical and Biophysical Research Communications, 2014, demonstrated that impaired autophagic flux in fibroblasts from older donors triggers alterations in extracellular matrix protein content and contributes to deterioration of dermal integrity and increased skin fragility. One of the consequences of reduced autophagy in fibroblasts is the accumulation of a specific cargo receptor, P62, also known as sequestosome 1, which builds up when autophagy is insufficient to clear it. Elevated P62 levels have been associated with senescent fibroblast phenotypes, and senescent fibroblasts are known to secrete a pro-inflammatory mix of cytokines, proteases, and growth factors, collectively called the senescence-associated secretory phenotype, or SASP. Research by Kang and colleagues, published in Science, 2015, established that GATA4, a transcription factor required for SASP activation, is normally degraded by P62-mediated selective autophagy, and that when this autophagic degradation is suppressed, GATA4 stabilizes and activates NF-κB-mediated inflammatory signaling, directly linking autophagic activity in fibroblasts to the inflammatory environment of the dermis. When autophagic flux in fibroblasts operates at elevated levels over a sustained period, the consequences for the dermis extend beyond protein clearance alone. The reduction in P62 accumulation, the continued degradation of GATA4 through selective autophagy, and the maintenance of lysosomal degradation capacity collectively shift fibroblasts away from the senescent secretory state and toward a functional profile characterized by lower inflammatory output. The dermis is, in this sense, only as functional as the intracellular machinery of the cells that build and maintain it, and that machinery runs through the autophagy pathway. After 72 hours of sustained autophagic activity, the changes that have occurred inside skin cells are molecular and structural, rather than visible to the naked eye in any dramatic sense. What the research collectively describes is a shift in the intracellular landscape of both keratinocytes and dermal fibroblasts, a reduction in the load of damaged proteins, dysfunctional organelles, and oxidized lipids, and a corresponding shift in the cell's capacity to allocate resources toward maintenance and synthesis functions. This is not a transformation in the colloquial sense. It is a measurable recalibration of the cellular environment documented across multiple experimental models and cell types. The scientific literature on autophagy and skin aging consistently points toward autophagic capacity as one of the variables that determines how well skin cells maintain their function over time. A comprehensive review by Eckhart, Chakler, and Gruber, published in Frontiers in Cell and Developmental Biology, 2019, examined autophagy across skin cell types and identified autophagic flux as a determinant of both epidermal renewal efficiency and dermal matrix quality, positioning it as a central mechanism in the biology of skin aging rather than a peripheral one, particularly in long-lived cell populations such as melanocytes and secretory cells of sweat glands that depend on it for cellular homeostasis. What 72 hours represents in this context is a window long enough for autophagic flux to progress through multiple cycles of initiation, cargo sequestration, lysosomal fusion, and degradation across the skin cell populations. The lysosomal compartment, which executes the final degradation step, turns over its contents on a timescale of hours, meaning that sustained autophagy over 3 days allows for repeated processing of intracellular material rather than a single clearing event. Researchers studying autophagy kinetics in mammalian cells, including work published in Molecular Cell, have described this iterative quality of autophagic flux as central to its biological impact. The benefit is not in any single cycle, but in the cumulative reduction of intracellular damage load that sustained activity produces. If you found the science in this video genuinely useful, hitting the like button helps this kind of research-based content reach more people. Subscribe if you want to keep going deeper into the biology. There's a lot more of this coming. And if you know someone who's interested in how the body works at the cellular level, sharing this video is the best way to bring them here.